Hello students, welcome to Bio Digest. Whatever makes you feel bad, leave it. Whatever makes you feel happy, keep it. With this beautiful quote, I welcome my students to the biology class. Today we are going to discuss about the structure of chromosomes. What are actually chromosomes? Chromosomes, the definition is, these are highly condensed coiled chromatin fibers. So these structures are actually found within the nucleus of a cell. Okay. So this is the nucleus. I'm just showing the nucleus of the cell. You know that it's having, this is the structure of a nucleus. In between you will be able to see nuclear pore. Okay. So this is the nucleolus, and here you will be able to see thread-like structures like this. These thread-like structures are what is called as chromatin fibers. These chromatin fibers, what they will do? During the time of cell division, that means just before cell division, some changes will be happening inside the nucleus. So these thread-like structures, they will condense. They will condense and condense, they will coil, they will be forming a structure like this, which is what is called as the chromosomes. Now coming to the discovery of chromosomes. Chromosomes were for the first time discovered by a German scientist known as Walther Flemming in the year 1882. He discovered chromosomes in the larvae of salamander. Salamander is an amphibian. So in the larvae of salamander, he was able to see certain thread-like structures. So that was about the discovery of chromosomes. Now we will see what are the materials that constitute the chromatin fibers. So the chromatin fibers are basically made up of two things. 60% proteins, which are actually called as histones, and 40% of DNA. That is deoxyribonucleic acid. You know what is the function of DNA? DNA is the genetic material. That means it is the DNA that will be transferring the genetic information from one generation to the next generation. Now, actually the structure is like this. I will be showing the structure. So, this is actually the structure of histones. Okay, you can see 4 ball-like structures are found in the front. And at the back also you can see 4 ball-like structures. So, in total you can see, I am showing the side view, you can see 8 ball-like structures will be there. Okay. So this is actually the histone molecule. We will be calling it as a core of 8 histone molecules. Core of 8 histone molecules. So core of 8 histone molecules will be there, and this will be actually surrounded by the so-called DNA. Okay, so the histones will be surrounded like this by the DNA. So this structure is what is called as nucleosome. So what is a nucleosome? A nucleosome is actually made up of a core of 8 histone molecules, which is actually spirally wounded by the DNA. That structure is what is called as nucleosome. So that means the structure is actually like this. Okay, you can imagine, I have already shown. So again, so this is the pattern. Okay, so this is how a nucleosome will appear. So in a chromosome, there will be millions of nucleosomes present. So this is a nucleosome. Now coming to the structure of DNA. The DNA is actually consisting of nucleotides. Okay. Now we are going to learn the structure of DNA. So what are the building blocks of DNA known as, or with which material DNA is actually made up of? DNA is made up of several nucleotides. So these are the building blocks of DNA. So now what are these nucleotides made up of? A nucleotide consists of sugar, phosphate group, and nitrogenous base. Okay, so this will be the structure. DNA is consisting of a pentose sugar. Pentose means this is actually a pentagon. 5-sided sugar. 1, 2, 3, 4, 5. 5-sided sugar will be there, which is known as pentose sugar. Then I told you it is having a phosphate group attached. So there will be a phosphate group like this. Then there will be a nitrogenous base present. So I am writing nitrogenous base as NB. Okay, for you to understand. So this structure is what is known as a nucleotide. So nucleotide, these are actually the building blocks of DNA, which is actually made up of a pentose sugar, a phosphate group, and a nitrogenous base. Now there are 4 nitrogenous bases. We will see what are they. Okay, so I am writing the nitrogenous bases here. Basically, the nitrogenous bases you can divide into 2. That is purines and pyrimidines. So purine bases are adenine, represented by A, and guanine, represented by G. And pyrimidine bases are cytosine, represented by C, and thymine, represented by T. Okay, so these are the nitrogenous bases. So nitrogenous bases are actually divided into 2, purines and pyrimidines. Which are the purine bases? Adenine and guanine, which are represented as A and G respectively. And the pyrimidines are cytosine and thymine, which are represented as C and T respectively. Now let us see what is actually the structure of a DNA. what is the shape of a DNA molecule. Okay, DNA will be appearing like a double helical structure like this. Okay, this is known as a double helix. So here this is actually similar to a staircase. Okay, we'll be calling this as a staircase model, or you can compare this to ladder. Okay, so this is the shape. So ladder or staircase. Staircase, you will be stepping onto the steps, isn't it? So here steps will be there. So what will be there here in this region? Here you will be able to see the nitrogenous bases. So if A is there, that means if adenine is there, opposite to that always there will be thymine present. So you can remember like at. Okay, so if A is there, opposite side there will be always T present. So adenine will be always binding to thymine by a double bond like this. So if thymine is there first, opposite what will be there? There will be adenine. That means A can always pair only with T by a double bond. So if T is there, definitely opposite side what will be there? There will be A present. That is adenine will be present. Now if G is there, opposite side there will be C. So that means guanine will be binding to cytosine by a triple bond. Okay, so difference is there. A and T, double bond is present. G and C, triple bond is present. Okay, so if C is there this side, what will be there on the other side? There will be G. So this also you can remember almost these two letters. C, if a child begins to write G, first the child has to write C. So like that you can remember. So C and G always bonds together. Okay, and what type of bond is there between? In between there will be a triple bond present. Okay, so like this bonding will be taking place, which actually gives a sort of code. Okay, a coded message will be formed. Now what will be there in this particular region? So here what I am shading in this region, this is actually consisting of the pentose sugar and the phosphate group, that is the backbone. Okay, so pentose sugar and phosphate group will be present in these regions. Whichever region I am shading, pentose sugar and phosphate group will be present. And in the step-like region, you will be able to find the nitrogenous bases. So this model is what is called as a double helical structure or a double helix. That means this is one strand, this is another strand. They are complementary to each other. So this is the structure of a DNA molecule. So we are learning about the structure of chromosomes. I told you what are chromosomes. Chromosomes are highly coiled condensed chromatin fibers. What all materials are present in a chromatin fiber? The chromatin fiber is actually made up of 60% of proteins, which are called as histones, and 40% of DNA, that is deoxyribonucleic acid, which is actually the genetic material. Structure of chromosome. So when you are drawing the chromosome will appear like this. These are called as the arms of chromosome. The arms of chromosome is having a name in biology. It is known as chromatid. So these chromosomes are having two arms. So if you are labeling both like this, you will use plural form, that is chromatids. Now this is the first arm. I am drawing like this. This one is the second arm. Both are belonging to the same chromosome. Okay. So if both the arms are of the same chromosome, we will call that as sister chromatids. Now you can see there is a particular thing that is present. This region is known as centromere. So question can come. What is the function of centromere? So how will you write the answer? Centromere will hold the chromatids together. That is the answer. Centromere, what is the function? It will hold the two chromatids together. Or you can write it will hold the two arms of the chromosomes together. That is the function of the centromere. So this is the structure of a chromosome. If you are asked to draw the structure of a chromosome, you just have to show the structure like this and label it like this. So sister chromatids and centromere. Okay. So that is about the structure of chromosome. So as we studied the structure of chromosomes, we will learn the definition of a gene also. What is the simplest definition of a gene? Gene is the physical and functional unit of heredity. This is the simplest definition. Physical and functional unit of heredity is what is known as a gene. So gene actually consists of DNA molecules. Okay, DNA will be present. I told you DNA is actually the genetic material. So it will be passing the informations, the genetic informations from the parents to the offspring. Let us recall. What are chromosomes? Chromosomes are the highly condensed coiled chromatin fibers. Discovery of chromosomes. Chromosomes in animals were first studied in 1882 by a German scientist, Walther Flemming. Structure of chromosome. Each chromosome consists of two chromatids, better called as sister chromatids, which are held together by a centromere. Chromatin. The chromatin material that constitutes the fibre is formed of two substances. 40% DNA, that is deoxyribonucleic acid, which is the genetic material, and about 60% of histones, a particular type of protein. The DNA strand winds around a core of 8 histone molecules. Each such complex is called a nucleosome. A single human chromosome may have about 1 million nucleosomes. Structure of DNA. The DNA is a very large single molecule. It is called as a macromolecule. It is composed of two complementary strands wound around each other in a double helix. Each single DNA strand is composed of repeating nucleotides, which are made of 3 components: phosphate, sugar, and a nitrogenous base. Please do subscribe Bio Digest. Like and share. Thank you.